Alright, my girls, so I decided to do the webcam plus the screen sharing because I thought you guys might want to see my face for the first time in a while. Maybe not, but you know, if you do, here it is. So I wanted to talk a little bit more about translation and kind of wrap up our lesson on how we go from the DNA to proteins. And I wanted to talk about mutations, which are changes that happen in the DNA sequence. They can be good, they can be bad, but just overall what mutations are, what are the different types, what are the consequences, what are the benefits of having a mutation. So we'll talk about that in the next couple of slides. Okay, go. Okay, go. Okay, it's not going. Okay, there we go. So just a couple of last minute things about the genetic code. I did talk about this at the end of the last video, but I wanted to touch on it again. So the genetic code, so our DNA, it's both redundant and unambiguous. So what do I mean by redundant? If we look to the right at all of our codons, there are going to be 64 total codons, and 61 of those are going to code for an amino acid. The other 3 are stop codons, which don't code for an amino acid at all. But there are 64 total codons, but we've already said in past videos there's only 20 amino acids, and they just get put together in all of these different combinations to make all of the different proteins that we know about. However, there are 64 codons, so those numbers don't match up. You would think, oh, well, if there's 20 amino acids, then there's 20 codons. There's not. There are 64 codons. 61 of them are considered coding because they code for an amino acid. So that's the redundant part. There's more than one codon that can code for the same amino acid. And then the unambiguous part would be the fact that one codon will only ever specifically code for that one amino acid. So for example, our start codon, AUG is always and forever only going to code for methionine. It's never going to code for anything else. So there's that. And then let's see some other ones. So I'm just making this up, CUC. So that's leucine. CUC will only and forever always code for only leucine. You see what I mean? So it's unambiguous. Redundant, but unambiguous. Okay, so I had to pause it for a second because Matt was rolling through in his wheelchair and he was making a lot of noise. So I can't exactly remember where I left off, but I think I left off being done explaining about what unambiguous meant. So the other bullet on this slide says that there are only 45 tRNAs. So remember, the tRNA is the RNA that's going to bring the amino acid to the ribosome because the tRNA has that anticodon that pairs up with the codon on the mRNA. And there's only 45 transfer RNAs. And you would think that since there are 64 codons, that each of those 64 codons should have its own tRNA. So you should really think, okay, well, if there are 61 coding codons that actually code for an amino acid, then shouldn't there be 61 tRNAs? Why are there only 45? And it's because of this really cool phenomena called wobble. And yes, scientists decided to name it wobble. We give all kinds of crazy names to everything, and sometimes those names are really difficult, and then sometimes we come up with stuff like codon wobble. Anyway, it's what it's called. But what is codon wobble? And I'll try to find like a shorter video that can kind of show you in motion how this wobble actually works. But basically the third base, so that final base of the codon, doesn't always have to match up perfectly between the mRNA and the tRNA. So sometimes the message on the mRNA might have like a T as its third base, and then instead of, or I guess it wouldn't be a T because there's no T in mRNA. So let's say it has like U as its third base, and then the tRNA, you would expect, since it's supposed to match up, it would have A because A is going to pair with U, right? except sometimes it doesn't. Sometimes they don't match up perfectly, and the transfer RNA will still bring the appropriate amino acid. So that's why there's only 45 tRNAs, but 61 coding codons for amino acids, because that third base doesn't always have to match up perfectly. So that's called the wobble. Not like the dance, but like DNA and mRNA. So I also mentioned RNA splicing in the last video, but in case it was kind of confusing for you, I have it here blown up just a little bit bigger. But basically it's going to give eukaryotes an advantage because eukaryotes, because we have a nucleus, we can't complete transcription and translation at the same time. Transcription has to happen first, and then that cap has to be added to the transcript, and that poly A tail. Everything has to happen before it can leave the nucleus to be translated. But in prokaryotes, they don't have that problem. Transcription and translation can occur at the exact same time because they don't have a nucleus to get through. So for eukaryotes, this alternative RNA splicing helps them in order to make a lot of different polypeptides or proteins from one mRNA sequence, because it can just pick and choose which exons it wants to include and which ones it doesn't want to include. So hopefully if you didn't understand it in the last video, this one kind of, now that you've had time to sleep on it, this one will make more sense. So now to talk about mutations. So it is a change in the genetic information of a cell or a whole organism, because remember some organisms are single-celled. We'll talk about really large-scale mutations when we get to meiosis. So what do I mean by like a large-scale mutation? That would be like how someone ends up with a third copy of the 21st chromosome and they get Down syndrome. We'll talk about those mutations when we cover meiosis. For right now, I just want to talk about smaller scale mutations. And so small scale mutations are ones that are going to affect between 1 and 3 nucleotide pairs, typically. Sometimes a little bit more than that, but anything much more than that is going to be considered a larger scale mutation. And I think I've said this before, but I want to reiterate, not all mutations are bad. People hear the word mutation and they just automatically assume, oh, well, a mutation is bad. But sometimes a mutation can be really beneficial. Like sometimes you have a mutation that gives you the ability to, I don't know, maybe smell something that's poisonous to you so you know not to eat it. Those are the types of things that are results from mutations. You currently, right now, as you exist, are the result of millions of years of good, favorable mutations. They're a main driver for evolution, which will be one of the last units that we talk about this year. Okay, so the first type of mutation I want to talk about is a point mutation. So that's going to affect only one nucleotide pair out of the entire DNA sequence. If a point mutation occurs in a gamete, and if you remember correctly, gametes are the cells that are responsible for reproduction. So your sperm cells and your egg cells, those are your gametes. If a point mutation occurs in one of those, then that can be passed on to offspring. But not all mutations get passed on to offspring, which is really cool. So what's an example of a point mutation? Have any of you heard of sickle cell anemia? If you have not heard of sickle cell anemia, I highly suggest you just do like a really quick Google search. Just pause this video and do a quick Google search, like what is sickle cell anemia, and really you can just read whatever pops up like on Google. You don't even have to click into anything. But sickle cell anemia is an example of a point mutation where a thymine nucleotide gets replaced by an adenine nucleotide. So in the whole nucleotide sequence, out of the millions and millions and millions of nucleotides that make up your genome, your entire existence, one change where this T gets replaced by an A, and thymine gets replaced with an adenine, that one single change causes sickle cell anemia. And essentially what's happening in that case is the protein would code for a valine amino acid where it would have normally coded for a glutamine amino acid. So basically you switch the amino acids out. And if you remember way back to biochemistry, and I had mentioned that your amino acids have different classifications. They can be polar, they can be non-polar, they can be acidic, they can be basic, they can be charged, they can be neutral. So there's these different classes, right? Well, valine and glutamine clearly are not going to have a similar property. They're not going to be in the same category, which is why if someone has sickle cell anemia, their red blood cells take on this sickle shape that I'm kind of pointing out right now on the screen. But they take on that sickle shape, which makes them not carry oxygen as effectively because there's not as much room. A normal red blood cell would look like this. Right, so that is a one nucleotide change out of the entire DNA sequence. That one nucleotide change in that one protein switches out that amino acid and causes you to have a completely different protein that gives you a completely different shape of your red blood cells. So that's kind of a drastic example, but you get the point that one small change can make a big difference. Okay. So what are some examples of point mutations? One of them would be a substitution. What I just talked about where the thymine gets replaced by an adenine, that's considered a substitution mutation. You're taking one nucleotide out and you're subbing another one in. That's a point mutation. Sometimes that substitution will code for the same amino acid. In that case, if it codes for the same amino acid, then you're not going to have any effect on the protein, and we call that a silent mutation. Okay, we call that a silent mutation. Because you coded for the exact same amino acid, therefore nothing happened. So unless this person was in the hospital or they were getting like genetic testing for something else, they wouldn't even know that they had this. They wouldn't even know they had this mutation because the protein structure is not affected at all. It's a silent mutation. But then sometimes you might code for an amino acid that is maybe similar. So like I was talking about on the last slide, how valine and glutamine are clearly going to be very different amino acids. They're not going to be in the same classification because when you have that substitution happen, you get sickle cell anemia, which is not good. You get this big change in your protein structure. But sometimes you do switch out for an amino acid that's really similar, that's in that same category. So maybe you change out one that was positively charged for another one that's positively charged. Or you change out one that was hydrophobic for another one that's hydrophobic. Since you're switching out for the same type of amino acid, you don't really have a big effect. You may have some small effect on the structure of that protein, but overall you probably won't have a big resulting change from that. And so we call those a missense mutation. So they make sense, they just don't always necessarily make the right sense. So it's a missense mutation. And then the last one is a nonsense mutation, and that would be kind of like what happens with sickle cell anemia. Or, well, I guess not really. I guess sickle cell anemia would be more of a missense mutation. A nonsense mutation is when you end up having a codon that would normally have coded for an amino acid, it's now a stop codon. And so your whole protein, once you reach that stop, that's going to signal to all of the machinery in your cells, okay, we need to stop making this polypeptide now. It's done. And so what's going to happen is you're going to have premature termination of translation. So the translation of that protein is going to stop way before it's supposed to. And okay, I have like a minute to get through this last set of mutations. So insertions and deletions, these are different from point mutations. Insertions occur when you add nucleotides into the DNA sequence, so nucleotides that were never there to begin with, you just like add in some more. Deletions is the opposite of an insertion. It's when you take out nucleotides. And normally adding or taking away nucleotides can be fine, like this insertion or deletion can be fine if you take it away in multiples of 3, because it takes 3 to make a codon. But if you start to take like 1 or 2 and that's it, either added or taken away, you can cause what's called a frame shift mutation. And a frame shift mutation occurs when the reading frame for the mRNA is offset a little bit. So if you look on the next slide, that sentence is supposed to say, the Blue Dog Cafe in Lafayette is delicious. But essentially, a frame should